Welcome to the Metabolic Link, a medical and science-focused podcast that explores the common thread of metabolism in health and disease. This is where science meets society. Welcome to another episode of the Metabolic Link. I'm your host, Victoria Field, here with my fellow co-hosts today, which is really exciting, Dr. Angela Poff and Dr. Dominic D'Agostino. Hey guys, how are you?
Hey, good. How are you?
Doing good.
Doing
Excited. Well. Yeah, no, I'm excited for another journal club. I know things have been quite busy for the two of you. Maybe give us a brief update before we dig in. So today's episode, just for those listening, is a journal club where we dive into the latest science, dig into papers, and have a great fun discussion around it. So if questions pop up from listening to an episode like this, I'm sure they will, because we're just asking questions and just having a great back and forth banter about them. Please pop them into the comments. Let us know what you think about these episodes. We always love feedback, because the more feedback we can get, the more we can give you what you want on this podcast. Be sure, if you like this episode, to leave us a comment, review, all the things, share it with your friends. And also know that we have a membership platform dedicated exclusively to metabolic health and therapy called the Metabolic Initiative, where you can actually find ad-free episodes, private episodes as well, but ad-free versions. So this video in particular, you'll find an ad-free version of it on our platform if you'd like to listen without ads. However, I will say supporting our sponsors that you'll hear more from on this episode supports this podcast and helps us make this happen. So keep that in mind, that we carefully vet the people that we work with. We love the brands that we work with, and we really hope that you'll join us in supporting them, because they support us. So without further ado, let's get back to you guys and tell me what's happening in the science world for the two of you, because I know in addition to doing this podcast, you're also full-time scientists and all the things. So Angela, do you want to get started with how things are going?
Oh, sure. Things are good. Busy as always. Dom and I just had a lab meeting a couple hours ago with the team, kind of getting some updates on a few of the projects going on in the lab. We're wrapping up a few different studies right now, which is exciting. One of our great graduate students just started medical school, so she was joining us by Zoom because she can't quit the lab, it seems. So we were excited to have her there. And yeah, and then I just got back from a conference in Chicago for the ACCME. Sorry, my cat's jumping up in the screen behind me, but just ignore her. And where that's the accrediting organization that allows us to offer continuing medical education credits to all of our healthcare providers out there who join us on the Metabolic Initiative. So that was a really great experience too, getting to hone in on ways to improve and expand our CME providership. So I'm excited to kind of put into practice a few of the things that we were talking about over the last days.
Yeah, that's really exciting for sure, because we want to continue to offer physicians medical education where you can come to our website and actually gain credit for watching the great videos on metabolic health and therapy. Briefly give us, for those who might be new to the YouTube channel or listening on their podcast player, what you guys do, your research lab, all of that, just for those who might not be familiar.
Yeah. Dom, you want to do that one?
Yeah.
Yeah, sure. Well, just briefly, what I've been doing is teaching.
Please.
So on the teaching front, been doing that, including mentoring students, including medical students on different projects, all related to metabolic health, continuous glucose monitoring, to sweeteners, non-sugar sweeteners, and things like that. Working on those projects, writing papers. We got a paper accepted today. I'm excited to share that pretty soon. On ketones and neuroregeneration or neurological repair. So historically, for people just like tuning in, our lab, I got to look at the date, for the last 20 years, I've been focusing on research associated with neuroprotection, and then that transitioned to a lab that does neuroscience and also cancer biology. And Angela pretty much took the reins on the cancer project, and that was 14 years ago, Angela, more than that, or about that time. And so we look at metabolic-based therapies as it pertains to primarily dietary therapies, ketone supplement therapies, some different metabolic-based drugs, monitoring systems and whatnot, and all things metabolic related, because we feel that metabolic health is sort of underlies our physiology, which we are physiologists and molecular biologists and biochemists, but that metabolic physiology is really the basis for our health and the prevention of the big things that's going to kill us, whether that be cancer, cardiovascular disease, Alzheimer's disease. So yeah, we've been kind of looking at all these different things in the lab in animal models, but we've also moved the science into human applications. So we've had clinical trials, one's at Duke University looking at ketogenic therapy for oxygen toxicity. And yeah, and we're working with Moffitt Cancer Center too on a couple projects that we hope will be funded in the near future.
That's really exciting. As research kind of drifts into human application and whatnot, that's really where we want to be, because as you mentioned, metabolism is the common thread that ties together health and disease. And it's exciting to see that. I feel like in the mainstream, what I've been up to is the podcast and the platform. That's my area of focus. So things that you see on the podcast. And then of course, we've got a great medical education platform that's keeping us busy as well. But it's really exciting to see the mainstream is starting to pick up on metabolic health as being this incredibly important, especially too, as of course, politics is all the buzz, not to get into politics at all on this podcast, but as we approach things like that, we're talking about like, what are the key issues in our country? Like, what should we be focused on? And this health or lack thereof of society right now, especially in America, is the number one thing we should be focused on, because if we don't have our health, what do we have, right? So anyway, long story short, it's exciting. It's an exciting time for the field. And yeah, so that's why we have these discussions where we can dig into the research, talk about it, put it out into the world. So if you're enjoying these types of episodes, let us know. So why don't we actually get into the research?
On that.
Oh, yes,
Yeah,
please.
on the,
Go,
like
Dom.
the politics, but so
Yeah.
well, listeners will hear the podcast with Todd Wagner, who has Food Fight USA, and we did have a discussion on that. This is a very nonpartisan issue. In particular, and it's been mentioned by him and several other people, are even heard today, that ultra processed foods are like cigarettes, basically, like today's cigarettes, and that is a major driver for and it took me, like, I was not completely convinced until I went down the rabbit hole the last couple of weeks that's a big player. But just from a political, not to get into politics, but this is like, everyone would agree that metabolic health, especially in the context of food safety, ultra processed food, and that could be sugar, even the chemicals in the food. So yeah, so no one would disagree with that. And I think that's an important
Yeah, exactly. I think that's a
is
good thing.
recognized. Yeah.
For sure. That's a good thing to mention, that we can all disagree or hate each other for hopefully not for different things.
Yeah.
But not on something that affects all of our health, especially the health of our children, right? That's something too that I think we can all agree on, that we care about. So at the end of the day, we need to be talking about this information. So without further ado, that was a good little intro there. Let's dig into some of these papers. We've got two papers today. One that is going to be diving into really focusing on different types of diets and their impact on glucose tolerance, lipid metabolism, and the gut microbiome, and then the other with a focus on cancer. So why don't we get started with the first, which is, I will read you the title: Ketogenic diet but not free sugar restriction alters glucose tolerance, lipid metabolism, peripheral tissue phenotype, and gut microbiome, RCT. And Dom, I think you're going to be, basically, for those who haven't heard this kind of an episode before, he's going to lead us through the paper, and then we're going to have a little discussion around it. And then Angela's going to lead off on the cancer paper. So Dom, take it away.
Great. Yeah. I have like the hard copy because I'm kind of old school like that, and I need it kind of in my hands. Yeah, this was a really good paper. It covered a lot of ground. And what I really liked about it, well, I follow Javier Gonzalez on Twitter. So everybody listening, follow him. He is really an expert in metabolic physiology, not just like, nutrition, biochemistry. And he has a high level of expertise in what I would call like understanding, he's from the University of Bath in the UK. And Aaron Hengst is also the first author, and they share the same lab. They're experts in metabolic physiology and really understanding metabolic flux in the context of macronutrient diversity, if that's a term, in the context, metabolic flux in the context of fasting, and metabolic flux in the context of exercise. And that's what I knew him for. And Christy Storashuk, our assistant, Christy, actually interviewed him. And that was the only kind of interview. Maybe he's done others, but I was familiar with his work, and he published this paper, and it had a few key things. I think the quality of it's in Cell Reports Medicine, which is a high impact journal, very well respected journal, looking at the methodology, looking at the methods and the level of data analysis, and even the graphical abstract, which Cell Reports requires, and a number of journals today require that. The graphical abstract has more information in it than I've ever seen in any other. Like, you could basically look at the graphical abstract, and I don't know if you can't see it there, but it has so much information in it. So I could just look at that and kind of talk about it today, but we'll go through some of the figures. But the highlights of the paper, I think, that are worth mentioning, and there are certain things within the paper that I feel were discussed ad libitum to a large extent, that I feel were not like translationally relevant. They're very interesting mechanistically, and they kind of maybe allow us to, ask some more hypotheses, but I'm going to really just focus on the low hanging fruit here in regards to the takeaways that people can get from this particular study. Again, the study name is Ketogenic Diet, but not sugar-free, not free sugar restriction, alters glucose tolerance, lipid metabolism, peripheral tissue phenotype, and gut microbiome. It was a randomized controlled trial. The highlights of the paper are that, just going there for highlights, and I have comments on their highlights, that low free sugar or carbohydrate intake reduces body fat, but not energy expenditure. And Dr. Gonzalez, Javier Gonzalez, is really focused on like energy flux and energy expenditure from the context of exercise physiology. And he's done some really elegant work with a number of different people in the field. I think he even collaborates now with maybe NIH on different projects. So there, a big takeaway was that what I think like inspired this, the question that they had that led to this study was that they were asking, would restricting sugar and would a ketogenic diet alter energy balance and metabolism, and would it alter overall energy expenditure in the context of activity? And that was sort of like one of the big questions that they had. And there was what they found was that there was weight loss, but there was no sort of metabolic advantage independent of calorie intake. And we can get to kind of that. They did find that restricting sugar lowers LDL-C and had a minimal impact on metabolism and the gut microbiome. That's restricting sugar. Unfortunately, I think some of the highlights, which Cell Reports Medicine asked people to do, to put like just 4 bullet points. One thing that strikes a lot of people and actually blew up my inbox was the 3rd bullet point on the highlights, which reads, 4 weeks of a ketogenic diet increases atherogenic lipid profile. But they kind of fail to mention that this was transient. So all it says is 4 weeks of a ketogenic diet increases atherogenic lipid profile. However, if you go to the data, that's not the case at 12 weeks. So there's a transient increase in LDL and ApoB, but it was like, it was like 10 to 15 points, but then it came back to non-significant after 12 weeks. So I got 6 or 7 emails about this, and then an exchange of emails that probably ended up being about 30 or 40 emails. One was actually from the Moffitt Cancer Center, and there's 2 groups there that were, hey, did you see the study? It's an RCT, and it's showing potential heart problems could arise. They just wanted sort of input on it. And it's like, hey, well, if you look at the data, in 12 weeks, it goes back to normal. So that's
Right.
something that we can discuss when we get to the data. And then another thing that I'm not going to talk too much about, but just go to the physiology, is that the fourth take-home message was the ketogenic diet alters gut microbial beta diversity and also skeletal muscle phenotype, which is kind of interesting. But I'm not going to talk any more about the gut microbiome, because they did see changes in the diversity of the gut microbiome, but they saw absolutely, like, after 12 weeks, no functional changes, nothing that would indicate a functional changes from the diversity in the microbiome. For example, they look at like gut permeability, and that did not change, and different aspects of that. So, so one thing that they did find that I'm surprised that they didn't put on it was that a low carb diet significantly increased GLP-1. So GLP-1 was significantly elevated relative to the sugar or the control diet and the sugar restriction diet. And that was significantly elevated. The ketogenic diet induced GLP-1 was elevated at 4 weeks and at 12 weeks. So I would have put that on there. I mean, with all the buzz of GLP-1s and everything.
Genova Connect, powered by Genova Diagnostics, offers simple, at-home, advanced lab testing that's grounded in research and takes the guesswork out of understanding your health. Genova's been pioneering advanced health testing for over 3 decades, and they have a wide variety of innovative testing options, including tests like Metabolomics Plus. This is a comprehensive nutritional test that's going to look at an individual's need for over 125 markers, including things like antioxidants, vitamins, minerals, amino acids, and fatty acids. Or you could try the comprehensive gut test, which is going to provide valuable insight into your digestive function, intestinal inflammation, and the intestinal microbiome. These are just 2 of the many options that they have available to help you gain in-depth insight into your health. So, whether it's gut health, hormonal balance, or nutritional insights, Genova Connect allows you to set a baseline and provide clarity along your journey to better health. And we want to thank Genova Connect for sponsoring this episode of the Metabolic Link. Genova Connect is offering you 15% off any of their test kits when you use the code Metabolic Link at gdx.net/themetaboliclink.
You're talking about the ketogenic diet versus free sugar restriction. And so maybe
not
kind of give
carb
us
restriction, but free sugar.
Yeah. So maybe explain for, those who might not know, because, the first thing you think of is sugar-free when they're talking sugar or free sugar restriction. What does that mean exactly? What, percentage of macronutrients comes from where? And then on the ketogenic diet front, how did that breakdown look for the ketogenic diet group as well?
For the macros. Yeah, I
Yes.
should have went. So the control diet, let's start with the control diet, was so they did equate for protein. So you had a control diet, a free sugar restriction diet, which had a level of carbohydrate that was 50% carbs. They just knocked it down to 5% sugar, as opposed to the control diet, more similar to what standard American diet, although somewhat healthy version of it, at 20% sugar. And the low carb diet, had like very little, like maybe 1 or 2% sugar. So you have a control diet that's on average of about 60% carbs, 30% fat, 35% fat, and then 15% protein. And that was compared to what they call a free sugar restriction diet. So just restricting sugar from, monosaccharide point of view, sucrose, down to 5%. So you have 45% of that sugar restriction diet was carbohydrates, complex carbohydrates, again, 15% protein and 35% fat. The ketogenic diet, on the other hand, was 77% fat. And again, standard, 15% protein, and then like 6 or 7% carbohydrates with like 1% of sugars, right? So that was, you have a controlled diet, and the graphical abstract compares the sugar restriction versus carbo, and then it goes down and it looks at a number of different physical activity, energy intake, body fat, fat oxidation, different lipid markers, fasting glucose. They use a CGM, which was very nice. Gut microbiome diversity, adipose tissue messenger RNA content, and skeletal muscle protein content. So these are the things that they look at, and I could go through each one of them, the results of that.
Yeah, absolutely. I think we should dig right in. And so thank you for that explanation, just because it could be a little confusing to those who don't have access to seeing the full graphs, but we'll be sure to link this in the show description so you can follow along with the paper if you're listening to it on a podcast player. But why don't we really dig into some of the weeds, because it was really interesting, maybe on the lipid metabolism front. I know you were talking specifically around how there was an elevation in atherogenic markers, only to see a decrease, I think, after the 12-week mark. This is really interesting because the shift in energy metabolism by following a ketogenic diet you can typically see this like kind of little increase, right? As your body starts to become adapted, and allows for it to transition into ketosis. And so this can happen for a lot of people, but then it kind of levels out. The other thing I think too, where you just kind of have that point around atherogenic particles, is it'd be interesting to see what happened to HDL. Let's talk a little bit about, I'm sure triglycerides tanked through the floor, because that's typically what happens. Some of these other markers that obviously give you the full picture of cardiometabolic risk is interesting. So why don't we start with kind of the initial bump that I'm sure you mentioned you got so many questions about initially in some of these atherogenic markers that they're taking a look at, and what that could be a result of.
Yeah, they didn't dive like too far into it. Unfortunately, it did make it into one of the highlights, but when you initiate a ketogenic diet, especially coming from the context of a high carb diet, it's going to cause some pretty remarkable changes in your blood work. So I think that's given. People will know that. And one of the things that is potentially atherogenic is the elevation of apolipoprotein B, and correspondingly the elevation of low density lipoprotein, LDL-C. So this went up statistically significant, it went up like to about 15 points. I believe I'd have to go to the graph, but thereabouts during the first 4 weeks when blood was drawn at the 4-week mark. Whereas just lowering sugar dropped, saw a relatively small decrease in ApoB and LDL-C, so a favorable response from just simply lowering the sugar content. But I think it's important to note that after about 12 weeks, there was no statistically significant increase in ApoB. There was it was still a little bit elevated, like 4 or 5, but it wasn't statistically significant. But there was, looking at the respiratory quotient, there was a significant elevation at 4 weeks of fat oxidation, was really high. I mean, if you look at the RER, and then that was obviously maintained throughout that 12 weeks. So you are doing a dietary intervention that's profoundly changing metabolic physiology in favor of fat over carbohydrate oxidation. So, and I think that's important to really think about and kind of conceptualize, because they did demonstrate that what they call glucose intolerance. So they talk about there was impaired glucose intolerance with the ketogenic diet. But one would expect that if you transition away from, and that largely results from things like a reduction in glycolytic enzymes, reduced AMP kinase, muscle PDK4, and the GLUT4 levels at the muscle, kind of like, immunohistochemically will get like internalized basically, and it's not at the surface of the muscle. So you don't get sort of that rapid translocation of the GLUT4 after you've been on basically a zero carb diet. So that's to be expected. And that was not really, people in the ketogenic community, in the exercise ketogenic community, know this, but I think that a little bit of a discussion could have, been a good idea there. So that was some negative things that were observed.
Hey, Dom, could you kind of make the argument, calling a glucose intolerance for someone on a ketogenic diet when you're not consuming many carbohydrates, you have insulin is quite low, comparatively speaking, you're not really primed, right, to be metabolizing carbohydrates in a way you are fats. You know, you could kind of argue, well, we could say that maybe there's a fat intolerance happening in the control diet here, or the free sugar restriction compared to the ketogenic, right? Because you have like huge amounts of fat oxidation in the ketogenic diet. But there's just kind of like this general, the way we kind of look at describing these kind of scenarios, and it doesn't really surprise me that, like, looking at this, you see this, you know, they would call this a glucose intolerance, but like, what about, could we call that a fat intolerance perhaps? Just, you know what I mean? It's just a different perspective and a different like focus, I guess, on, you know, the two different coins of the metabolic kind of scenario.
Yeah, that's a good point. And I think carbohydrate restriction and eating more fat, well, through carbohydrate restriction can cause a rapid reversal of fat intolerance, by definition, like elevated triglycerides, you know. So that is acknowledged and kind of observed. And I think, you know, that needs to be appreciated more. And then, you know, exercise can also, so their big reason for doing this was looking at physical activity, and they did not really see any changes there. But I think it's important to note that they did see pretty significant changes in reduced calories with the ketogenic diet group. So and they didn't really approach it from the perspective of reduced palatability. So there was a significant reduction in total calories. There was significant weight loss, significant fat loss with a ketogenic diet. And so body fat went down quite a bit, and energy intake went down. And they mentioned kind of briefly the GLP-1, and the graphs show that it was significantly elevated. But one could hypothesize that it was maybe reduced palatability, or not having a hyperpalatable diet, and then also the effects on the incretins, including GLP-1, I think factored in there, because protein did not change. That was equal across the group, so it wasn't like a high protein effect. And I think from it, the data meshed really nicely with data that we published, and Andrew Kutnick was the senior author, and Philip Prinz was maybe the senior. Andrew was, Philip, I think, was the first author, with a study where we looked at low carb versus high carb in a crossover design. And they saw something very similar to what we saw, which was basically just elevated glucose in the carbohydrate group. I guess maybe more appropriately would be that there was reduced fasting glucose. And if we look at the continuous glucose monitoring and fasting glucose, that was significantly reduced in ketogenic group. However, they did kind of they did show post-meal glucose in the carbohydrate group. And they kind of emphasized that, I think, and that's even in the graphical abstract, went up. And that is to be expected. And I don't think that's, you know, impaired glucose tolerance should be perceived as something negative if you're not eating glucose. That's to be expected. But they saw, you know, pretty remarkable enhancement of or reduction in fasting glucose and circadian glucose, if you want to kind of use that term, but an increase in postprandial glycemia with a carbohydrate meal, you know, if you're not eating carbohydrates. So that was observed, and I think would be kind of expected. So they saw lipoprotein lipase went up, like fat oxidation enzymes went up. And they did at the level of the skeletal muscle. You know, there's a reduction in AMPK, GLUT4, and a few other factors that could explain that.
I'm excited to share a really neat scientific breakthrough that supports our long-term health and wellness. Fatty15 co-founder Dr. Stephanie Van Watson discovered C15 as the first essential fatty acid to be found in over 90 years. And she did this while working with the US Navy to improve the health and welfare of aging dolphins. It's pretty simple. Essential nutrients are just that. They're essential for our body's cells to function properly. But we can't make them ourselves, so we have to get them from our diet. But that's not always easy to do. Fatty 15 is the world's first and only science-backed, patented, award-winning C15 supplement. Research suggests that C15 works in multiple ways. It repairs age-related damage to our cells, it protects them from future breakdown, it boosts mitochondrial energy production, and it activates pathways in the body that help regulate our sleep, mood, and innate repair mechanisms. And beyond supporting metabolic health, another feature of this product that I really like is that it comes in a reusable glass and bamboo jar with refill capsules that are shipped to your door quarterly in pouches made from recycled materials. Fatty 15 is on a mission to replenish your C15 levels and restore your long-term health. You can get an additional 15% off their 90-day subscription starter kit by going to fatty15.com/metabolic link and using code metabolic link at checkout.
Yeah, let's talk about the gut microbiome front, because it's really interesting that they measured that just in a little bit more depth, because I think it's kind of interesting the difference between the two. You've got, there were fairly significant changes on the ketogenic diet side, but maybe not as much on the free sugar side. So what does that tell you, possible long-term effects of that? There's so much unknown when it comes to the gut microbiome and what it really means, and extended amounts of time on any particular diet, I can only imagine might not be ideal for, gut diversity or what have you. But curious of either of your thoughts around that specifically.
It looks like they partnered with a company that really specialized in this. So they did have a lot of data on the gut microbiome, and they did show a reduction in microbial beta diversity, which could, in theory, if followed for a long periods of time, alter the gut in ways that could potentially be unfavorable. But there's so many unknowns to that. And I think there's compensatory things that happen. I think there's also a reduction in, even things like small intestinal bacterial overgrowth, or SIBO, which we have like an abundance of bacteria that could be causing some problems like, bloating and things like that, which kind of go away. I think the important thing, I spent quite a bit of time going through the microbiome data, and I didn't come up with any kind of conclusion as to whether it was positive or negative, even though there was a reduction in diversity. And I guess that would mimic their just going down the rabbit hole of different, like, what does this mean? What does that mean? And I didn't find any kind of meaningful takeaways from it. And they did not find any meaningful functional takeaways either. And I think that's really important. If someone brings this paper up and says, look, it's the ketogenic diet is bad for the gut microbiome because it decreases microbial beta diversity, but they will say, if you read into the paper, we saw absolutely like no functional changes in like gut permeability or other things that would correlate to that. They did see a transient increase in HSCRP, and they kind of harped on that a little bit as being atherogenic. But again, if you're on a high carb diet and you radically change to a high fat diet, ketogenic diet, that can induce things like even changes in redox system. It can activate NRF2 and things like that. Like you can get a bump up in oxidative stress that actually had been shown in some animal models. And then that transient oxidative stress can actually be a trigger for endogenous antioxidant systems, and also a trigger for beneficial adaptations later on. And they did see that there was a transient increase in CRP. I would like to mention that I've been on a ketogenic diet for a very long time, and my HSCRP goes between 0.1 to 0.2, and sometimes is non-detectable. So, and it only goes back up if I start eating carbohydrates. So everybody's different, but I'm just throwing my little N of 1 out there. And, but I actually have seen people go on a ketogenic diet and send me blood work, and I've seen their CRP go up. But that usually comes back down, and that would kind of mimic what they see in the paper here.
Yeah, it's interesting. I feel like that a big shift in diet like that. I mean, I feel like the ketogenic diet is more than just a diet. You're just going through a massive metabolic shift and change. So that in and of itself, like, compared to somebody who just cuts out sugar, right, and is on relatively similar macronutrient ratios, they're not, now just shifting over to producing ketones and that whole thing, but just
Mhm.
cuts out sugar, that change in gut microbiome is obviously not going to be as dramatic as somebody going from a standard American diet to a ketogenic diet, right? So
Yep.
that in and of
Yeah.
itself could explain that.
And I wonder, I don't know if you, I didn't look, did they report differences in fiber between the two diets? Because you could
Right.
imagine the ketogenic diet might be quite a bit lower in fiber, and that could explain some of the shifts in the microbiome.
For sure. Were
Yeah,
they like carnivore keto, or I'm kidding?
they were very low carbohydrate. Just looking at the total macronutrient, ratios and things like that, it would be less fiber for sure. Yeah, and that
Yeah.
could, decrease in soluble fiber, we know contributes to things like butyrate and some other, beneficial microbes. I just came from a talk where they talked about the potential reduction in butyrate, but beta-hydroxybutyrate can serve the same function at the colonocyte, and in circulation, your colonocytes have access to the capillary bed. So the beta-hydroxybutyrate that's elevated, you can make on a really engineered high fiber diet up to 50 grams of butyrate per day, I guess, from short chain fatty acids. But your body can make like 50 grams of beta hydroxybutyrate per day on a therapeutic ketogenic diet. And then that could serve the same function from the perspective of being a fuel for the colonocytes. And so that has been shown before. I just came from a talk where they showed that beta hydroxybutyrate on a ketogenic diet that's devoid of fiber can maintain the wall of the gut in a way, in the colonocytes, just through the capillary delivery of that.
This episode is brought to you by Maui Nui Venison. Maui Nui is helping solve a critical environmental problem in Hawaii through wild harvesting of the axis deer population that are invasive to Maui, overpopulated and damaging the ecosystem. Maui Nui offers the only 100% wild harvested meat that's completely stress-free to the animal and responsibly sourced. If you're looking for a snack that's delicious, healthy, and makes a difference, you've got to try Maui Nui. I keep Maui Nui meat sticks in my purse for a convenient and delicious high-protein snack. My husband has also become a bit obsessed with them. As an ER doctor, he spends long days on his feet and doesn't always get to sit down for a full meal. That's why he loves to keep these around for a tasty and filling source of protein. There's a no gamey taste, just savory and satisfying. Packed with essential nutrients, vitamins, and healthy fats, each stick contains 10 grams of protein at just 55 calories. That's up to 53% more protein per calorie than grass-fed beef. For Metabolic Link listeners, Maui Nui is offering 15% off your first order. Supplies are limited by the nature of their work, so secure your Maui Nui now by going to mauinuivenison.com/metabolic link and use promo code metabolic link at checkout. That's mauinuivenison.com/metabolic link and use code metabolic link for 15% off your first order.
People are always looking for key takeaways of like, okay, well, which diet do I follow after reading research like this and what have you? And it's so hard to take that from something like this. And obviously more work needs to continue to be done. In terms of body fat, it looks specifically because, everybody's looking to lose body fat, right? It looks like both of those diets, as I think you mentioned, Dom, help people lose body fat. This means cutting out sugar, simply cutting out sugar, or maybe going on a ketogenic diet could be helpful with something like that. And I just think overall, it's just really interesting to see some of the changes that happened. Something like gut microbiome, it's hard to say what we can take away from that. Same with like on the lipid marker thing, we're kind of not really seeing the forest through the trees when you're just talking about LDL levels, bad cholesterol, as they call it, when you're not focusing on other areas of, that contribute to atherogenic risk, like systemic inflammation, triglycerides, all of those other things. So I think there's some really interesting points that we can take away from this, but, just having going one way or another, or just, swinging the pendulum one way or another, and saying this diet is superior to that diet, we're still not there yet with that, I would say.
I think, well, I was just going to mention real quick that in getting to the atherogenic lipids that they talk about in the abstract, just knowing the authors and looking at some of their prior work and knowing what reviewers ask authors to do, I had a hunch that the mention of atherogenic lipids in the abstract, they may have requested that term used. Might have been requested by a reviewer to just throw it in there because just the way that it's mentioned, and not even mentioning that it was a transient, increase.
Right.
So just yeah, I know how they, but I think the take-home message here was that the data just reading the data suggested that the ketogenic carbohydrate restriction may not be necessary to produce cardiometabolic health benefits that would be expected by the weight loss observed alone, right? That, in other words, just free sugar restriction may be more appropriate dietary choice for overall cardiometabolic health. That's sort of their takeaway. Neither type of dietary restriction appears to meaningfully alter energy expenditure. So that was in a nutshell kind of their takeaways. Not poo-pooing the ketogenic diet, but just saying that it's a rather extreme approach that is not, well, they said could not lead to an unfavorable cardiometabolic consequences. And then just throw in that, reducing sugar is kind of the way to go for cardiometabolic health.
Yeah. Yeah. And for those who are listening who want to dig into additional research around cardiometabolic health, specifically in the ketogenic diet, there was recently a paper just released, really exciting, titled Carbohydrate Restriction Induced Elevations in LDL Cholesterol and Atherosclerosis, written, one of the folks on that is Dave Feldman and Nick Norowitz, as we've just recently have Dr. Nick Norowitz on the podcast. So paper I'd highly suggest checking out if you're interested in just better understanding. This is a step in the right direction, right? It's not the end all be all answer, but better understanding what's happening, when it comes to markers, cardiometabolic markers when you're on a ketogenic diet. Peel back the layers and take a look at actual scans and more data. It's really exciting to see this paper come out. So I'd highly recommend checking it out. We'll be sure to link it in the show description as well. Angela, were you going to say something too?
Oh, I was just going to say, we were talking earlier about the transient unfavorable lipid profile. And
Right.
I mean, that's something that has been reported in the epilepsy literature for like decades.
Right.
So like we know it goes, and in a lot of people it does kind of go in a negative direction for a little while and then kind of stabilize over time. So, it would have been nice to see a little bit more of that nuance there in the highlight. And since I think that's a really important part of that story. And it's been reported, we know this from other literature. So
yeah, when you don't include that context for anybody who's looking at a paper like this who doesn't understand that, or the history, or what else is out there in terms of research, they might automatically be sending Dom emails saying this is estrogenic, like what to explain that, understanding it's a transient thing. And anyway, so yeah, that context I think is really important as well. Super exciting paper. We'll be sure to link that one in the description, but we're going to shift gears to cancer metabolism. Angela, you are going to lead this paper off for us. If you want to give us the title and dig into the weeds, and then we can have a nice little discussion around this.
Yeah, absolutely. So this was a really cool paper, came out mid-2024, titled Ketogenic Diet Reshapes Cancer Metabolism Through Lysine Beta-hydroxybutylaration. And I'll apologize if I constantly trip over that word. So
I hate that word.
a few different ways.
That to you?
I've been saying it for years and it's still like, I don't know.
It's not my favorite word. Yeah.
Yeah. And this was published in Nature Metabolism, which is a great journal, and always cool to see papers like this in journals of that caliber. And also, there's going to be a heck of a lot of data in it when it comes out in one of the Nature journals. So, okay. We're obviously talking about the role of ketogenic diet, trying to kind of dig into mechanisms. There's been a lot of interest in ketogenic diet and cancer research for a long time. And, over the past decade or so, people have really started to kind of unravel these different, distinct mechanisms at play that kind of help explain a little bit more why we're seeing some of the effects we're seeing. And so this is referring to something called lysine beta-hydroxybutylaration. And what that is a type of something called a post-translational modification. So if you put on like your high school biology, thinking cap, and you think back to when you were learning about some of those basics of like biology, you have your DNA, which is like your blueprint for how you build all of the proteins that are in your body. So you have your DNA that gets transcribed into RNA that gets translated into protein, which are these chains of these amino acids. And so when you have a protein that's built at the end of this process, after it's been translated, that's not usually the final step. Even though you have a protein, there's a whole bunch of other like modulation that can happen after the protein is made that you can use to regulate the activity or function of that protein. And there's a whole host of different ways you can do that. And these are collectively called post-translational modifications. So modifications you make to the protein after it's been translated. So this is often like attaching or removing a chemical group, and this is going to affect how that protein functions. It might activate it, it might deactivate it, it might alter how long that protein lasts in the cell, how it is able to interact with other proteins or other biomolecules. And so it's this really broad kind of set of processes to regulate protein function. So you've maybe heard of things like phosphorylation, which is when you add a phosphate group to a protein, and that can change its function, or glycosylation, which is a sugar molecule that gets added, et cetera, et cetera. There's a whole bunch of these different things. So there's kind of like this classic set of the most common ones that we know about that I remember being taught back in the day. And then we've continued to uncover a whole host of new ways that you can post-translationally modify and control, regulate the activity of proteins. And really over maybe the last like 8 to 10 years, researchers have discovered one new type of post-translational modification that is highly relevant to our field, which is beta-hydroxybutylaration. So you know the word beta-hydroxybutyrate, that's that primary ketone body that we talk a lot about. And that is the primary ketone in the blood when someone's in a state of ketosis. So with beta-hydroxybutyrate, you have an addition of a beta-hydroxybutyrate molecule, so a ketone molecule, onto a protein to modify its activity. And the throwing in of lysine in front of that just means that's the amino acid that it attaches to. That's all it means. So in this paper, I said they were trying to kind of increase the understanding of these mechanisms of ketogenic diet and cancer. And, if you've listened to this podcast or followed any of our work, we have a really big interest in how ketogenic diet can alter cancer metabolism in a way that could potentially elicit therapeutic effects. So in particular, we're interested a lot in kind of going beyond this superficial idea of like ketogenic diet is, starving cancer of glucose. That's like a superficial, minimalist kind of view of all the things that happens in the context of physiologic ketosis as it concerns to cancer in the tumor microenvironment. And so there's so many different mechanisms at play. And one of those areas that's of major interest is looking at how ketones like beta-hydroxybutyrate themselves can interact with a cancer cell and change its activity in a way that could be anti-cancer. So, okay, we're talking about this new kind of process called beta-hydroxybutylation, and this can happen to both histone and non-histone proteins. So if you remember what a histone is, it's a protein that your DNA wraps around, and it basically helps control what genes are expressed. And then the other proteins just out and about in the cell and doing their thing, those would be like non-histone proteins. So Beta-hydroxybutyrate, we now know, can be added onto all of these different kinds of proteins within the cell. And this is all very, very novel. We're only really learning, when and how and why this is happening. So in this study, they wanted to look at that role of, okay, is the ketogenic diet maybe working in part through this newly described signaling mechanism for beta-hydroxybutyrate? Are you seeing when ketones are present, are they being added to proteins within the cancer cell or tumor microenvironment? And is that causing some of the anti-cancer effects that are being reported? So they really wanted to kind of take a really broad look at all of the things that are happening within the cell, within a cancer cell in the context of ketosis. And so they took 3-month-old mice, which is like a young adult mouse, and put them on ketogenic diet for 9 weeks. Mice only live like 2 to 3 years, so 9 weeks is a pretty significant amount of time for their lifespan. And as expected, you saw ketogenic diet mice, they reduced their body weight by about 5 to 10% compared to the control mice. They entered a state of ketosis, so their glucose was reduced and their ketones were elevated. And then the researchers did a whole host of multi-omics analysis. So transcriptomics, which is looking at the complete set of RNA that's being produced in the cell, proteomics, looking at the complete set of proteins that's being produced, metabolomics, so that all of the metabolites that are being made, and then what they call beta-hydroxybutylaromics. And I did have to practice that word earlier, which is the complete comprehensive study of where in all of the proteins in the cell are you seeing beta-hydroxybutyrate getting thrown onto a protein. And that set of data would be the beta-hydroxybutylaromics. So even though this is a really new process, there's been a lot of, we know that there's a bunch of potential binding sites for beta-hydroxybutyrate onto proteins. But to date, not very many of these have actually been studied to the point where we know that they actually are functional and have like real functional consequences. So one of the other cool things about this paper that I won't get into like very in depth because it's just a lot of detail is they went a step further and actually developed this hierarchical learning framework that allowed for the prediction of functionally important lysine beta-hydroxybutyrate. So not just, okay, it can attach to a protein at this site, but does it actually have an effect once it does? And in the paper, they were able to validate that several of these sites that they were looking at were functionally important as well. Okay, so what did they find? Well, they found that there were multiple metabolic pathways that are really critical for tumor initiation and progression. Things like glycolysis and TCA cycle were markedly reshaped by and affected by beta-hydroxybutylation. So massive changes in proteins that are serving these functions. They found almost 3,500 peptides that were exhibiting beta-hydroxybutylation. That's a really big number. And when they mapped those to their corresponding protein sequences, that resulted in over 5,000 unique lysine beta-hydroxybutylation sites and over 2,000 proteins. So that included over 4,000 of these sites that had not ever been reported before. So when you kind of take a step back and think like, oh my gosh, like understanding the scale of how many proteins can be impacted by this really important, post-translational modification that we didn't even know about like 15 years ago. It's really cool. And it's like a super exciting time as a scientist to be in this field and kind of seeing this really new understanding of how ketosis could be impacting something like cancer. So really what the biggest finding of the paper and what kind of they hone in on across the board is that they showed specifically that either ketogenic diet consumption or just treatment with beta-hydroxybutyrate, both of these, increased beta-hydroxybutylation of a particular metabolic enzyme called aldolase B. And it did this at several different lysine residues, so like several lysines within the aldolase B enzyme. And aldolase B is a known regulator of glycolysis and also mTOR signaling, which you've probably heard about. And so they showed that when this happens, and specifically at one lysine 108, you see reduced binding of aldolase B to its substrate, which is fructose 1,6-bisphosphate for all the biochemists out there. And this causes glycolysis to be downregulated and mTOR pathway signaling to be downregulated, which is awesome when you're trying to pump the brakes on cancer metabolism, because both of those pathways are heightened in most cancers and are really helping contribute to that cell's survival and proliferation. And so they coupled those findings with a series of really elegant studies, clearly demonstrating that taking that beta-hydroxybutyrate, butyrate, attaching it to those lysines on aldolase B was responsible for reducing the mTOR activity, which caused an inhibition in proliferation of these multiple different cancer cell lines. So not just one type of cancer cell. They looked at liver, kidney, gastric cancer cell lines, and they looked at several lines under each category. Okay, so again, we want to see that this is happening across the board, or at least in several, cancer types, which is super exciting. And so they did a whole bunch of studies and showed that through this mechanism, these cancer cells were less viable, they were dividing more slowly, they had reduced migration, they had less colony formation potential. And it was just really, really cool to see that level of mechanism in being described in this field. And it's this kind of data that's really going to perk up the attention of, people at the National Cancer Institute looking at funding this kind of work. This is the type of data that we've needed for a long time, and we're seeing more and more of. And the last thing I'll say is the paper is absolutely massive. I started to print it and it was going to be 50 something pages. So I decided not to print the reference section. So I saved 20 pages there. But they spent a lot of time showing all of their multi-omics data and validating that predictive beta-hydroxybutylaromics system that they were talking about. And I won't go into the details, but they used that system and they integrated known information on protein-protein interactions alongside their experimentally validated results. And they identified dozens of other proteins involved in these really important cancer metabolism pathways that could also be influenced by lysine beta-hydroxybutylation of aldolase B. So the take home there is that this specific event functions in a lot of really important biologic processes, and in particular, cancer metabolism, and it's a very exciting therapeutic target.
That was great. I'm glad you reviewed this and not me. I did print the whole thing out. The first thing I did when I printed this out is looked at all the work, and I looked at the funding sources. I think they had about a half dozen different sort of grants and funding agencies supporting
it. It's huge.
This is easily a $10 million project. It seems like, yeah.
Yeah.
Wow. But the research and the science, and I was excited to kind of go through it, and I still need to dig into this, and the references relates to Sarah's work and our work looking at BHB-lation. I like to just say BHB-lation.
That's probably better.
Beta-hydroxybutylation. But yeah, I mean, it's the results here are super compelling. The data looks really impressive, especially some of the Western blots and some of the mechanistic work. It was sort of when this whole idea came out, I was a little bit skeptical of, direct histone modification from ketone, but this seems to support a growing body of evidence to show, ketone-induced epigenetic regulation is a real thing. And I think, this will be an emerging area of research that a lot of people will be jumping into. So, and this will be a seminal paper probably in the world of cancer.
Yeah, totally.
Yeah. Really exciting paper for sure. Curious of your thoughts around, if so the mice rather were fed a ketogenic diet, if the effects that they observed could be replicated with maybe supplementing with ketone bodies, right? And
Yeah.
without a full dietary restriction, what that would look like. You guys have obviously done a lot of research around ketones specifically, so curious to your thoughts on that.
Yeah, they actually did both. And
okay.
the majority, so a lot of the work was in cell lines, and so it was just BHB like in and of itself. And then, but yeah, where they were looking at ketogenic diet and BHB application, they're seeing the same thing. So this is really, they nailed it down to this specific role of BHB and its ability to modify these lysines. And from this data, it doesn't seem to matter where that BHB came from, which is super encouraging in the field where, you know, there might be patient populations that are uninterested or unwilling for some reason to follow a ketogenic diet. And maybe in some cases, it's not even necessary. Maybe you can get those benefits through oral delivery of ketones, and that could be important for certain people for sure. So
right.
Really
well,
exciting.
Yeah. That's what I was thinking, you know, because it can be really difficult to implement a ketogenic diet with a cancer patient. At least somebody upon initial diagnosis, maybe they just start supplementation and it could have some of the same effects, which would be great. You know, beyond just cancer, right? Looking at folks with Alzheimer's and you name it, just seeing some benefits there.
Yeah.
And it's also as like, just for me personally, like we published a paper, I guess probably 10 or 11 years ago, looking at just the broad scale of ketones and you know, in cell culture being able to reduce viability and proliferation of cancer cell lines. And at that time we weren't digging into the mechanisms, but we're seeing this very clear, you know, reduction in their proliferation. And it just seemed odd. Like I remember taking that data to a bunch of conferences and people just kind of being like, what? Like, why would that's an energy molecule? Why would that, you know, reduce their survival? And well,
I just found that.
Yeah.
You know, I did a search, or no, I had to like register for some platform, and it says it throws all the stuff that you published, and it was like, are you an author of this or that, or was that whatever? And it brought up the precursor to the International Journal of Cancer paper that you published, Angela, which is entitled "Ketone Supplementation Reduces Metastatic Cancer" and some blah blah blah. But it was your first presentation, I believe. It was published in FASB Journal. The abstract was at Experimental Biology, and I think that's a 2012 or maybe 13 somewhere. I think that paper got published it was about a year or so before the paper. So we had the preliminary data of just ketone supplementation independent of the diet. You know, and you had the cell work, and I think you had some preliminary work with and it was racemic. So this brings up another question. We use the D and the L. We use racemic, and it's unclear what they used here, but the general consensus is that the D form of beta-hydroxybutyrate and the L form of beta-hydroxybutyrate, which our body does make a little tiny bit, it's about 2 or 3% in the tissues, they both have epigenetic signaling effects. So the implication of that is that if you consume a DL beta-hydroxybutyrate, the D gets burnt as fuel very quickly, and then the L stays in circulation about 2 to 3 times longer. So you could have, by taking a racemic or L, and I see a ton of patents now on the L, which we sort of predicted this was going to happen because I was talking about it a lot on podcasts, that the L could be like the ketone therapeutic. If we're talking about metabolic control of epigenetic regulation in the context of cancer therapy, a racemic beta-hydroxybutyrate formulation or even an L-enriched formulation could be a formulation that's superior or far superior from the context of activating this epigenetic, signaling cascade that has an anti-cancer effect. So maybe even away from no one's done that study yet, but DBHB is fuel, and it kind of can be used potentially as fuel where the L does not, is not favorably, is not oxidized. It's kind of oxidized. It goes through like kind of the same as fatty acid oxidation pathway. But I think there's a lot to be studied there of the two different enantiomers. And that seems to be a direction that the science is going to try to understand the fuel versus the signaling effects of beta-hydroxybutyrate. And this paper lends to that sort of whole idea of the signaling.
Mm-hmm. Yeah, totally.
Well, this paper for me brings up the thought around what this could mean when it comes to personalized medicine and, clinical translation and stuff like that. So I'm curious if biomarkers related to KBHB modifications could be developed to potentially like identify patients who would maybe benefit from a ketogenic diet-based therapy, more than somebody else, right? Is there something that could be developed? Curious of your thoughts around that, because that could be really helpful in pinpointing and offering personalized medicine for cancer patients, right?
Yeah, absolutely. I mean, you could envision getting to the point where, similar to how, in today's world, you can see what specific mutations someone's tumor is exhibiting, and you know that those pathways are particularly important, and therefore you can give, targeted chemotherapies. You could also be looking for markers that indicate that certain pathways that we know to be targets of beta-hydroxybutylaration, if those are upregulated in this patient's tumor, they might be a candidate, right, for ketogenic therapy. So I think that's a really good point. It's definitely a possibility, and probably a inevitable, down the road with just everything that we're seeing, getting the ability to have that kind of precise information and actually incorporate that for an individual based on their specific tumor. Would be excellent. And back to what Dom was saying, like there's certain cancer cells that might be more adept at taking in and maybe metabolizing DBHB for energy, right? Would they be more of a candidate for something that's more of like an LBHB focused therapeutic, where you're getting the signaling effects, but maybe you're not fueling from an energetic process? So there's a whole wide field open, I think.
Yeah, really, really exciting on what it could mean on the precision medicine front. What are some of I know we've talked about this with some other research, but what are some of the potential like challenges, I guess, and things to consider when you're trying to translate findings like this to clinical practice, right? Because a lot of people will read this, get really excited about all the possibilities, but there's still a pretty wide gap between the two.
Yeah, I mean, it's definitely, I think, for the major implication for the field is it just continues to validate mechanistic level understanding that ketogenic diet can have a major impact on these important molecular processes. I think that, to me, it's mostly contributing to the wealth of knowledge in a way that increases our confidence that this is a very valid approach, and gives people that maybe 10 years ago were saying, there's some anecdotal reports, or yeah, maybe in mice you see something, but like, I'm not convinced, when you can say, look, these are very specific and elegant mechanisms that have been clearly outlined. People start to pay attention, money starts getting thrown towards those directions, so that you can actually do the trials that need to happen. So it's from a perspective of like immediately like causing some translation into humans. I don't think it's necessarily at that point, but it is laying that really important groundwork that's going to, just as a whole, lift up the whole wealth of this research and push it forward even faster.
Yeah, it's definitely like a really great step in the right direction, the direction we want to be going for sure, and serves as a great foundation for moving forward. Well, thank you, Angela, for first of all the explanation and just how you weave in and out of the paper and provide just breakdowns of everything is just really eloquent and great. it's like you've been a teacher or something like that over the
I
years.
do like to teach.
Yes.
I like to teach.
It's like you like teaching or something. No, it was really great. Well done on that front. And same with you, Dom. Both of those papers, really, really fascinating. And we've linked both of them in the show description. So be sure to check them out, read them for yourself. Let us know what you think. Leave some comments for us. We always want to have a great dialogue. We're across all of your major social media channels. You can find us at Metabolic Health Summit or the Metabolic link. And yeah, we so appreciate you tuning in to this episode. We're going to have more journal clubs in the future. We release new episodes of the podcast every other Tuesday, so be sure to stay tuned. And I want to mention again, definitely check out our medical education platform, because if you really like to get into the weeds on this, we've got such an incredible, robust bank of presentations from our scientific conference, Metabolic Health Summit, over the years, from the very beginning, including the ones from 2024. And we're going to continue to release new presentations from actually outside of our scientific conference too. So be sure to stay tuned for that. We also have all of our podcast episodes up on the platform, ad-free there if you want to check it out. And also do great live Q&As that Dom and I host from time to time with great experts, which has really been a lot of fun to, you can hear some of these experts talk on the podcast, and then you can jump into a live Q&A on the platform to actually ask them your questions firsthand, which is a great, unique opportunity. So head on over to membership.metabolicinitiative.com to check out the platform. And guys, thank you so much for jumping on today to dig into these papers. It's super fun and so great to see you both.
Yeah,
You
thanks
too.
for hosting. Yeah.
Awesome. Thanks, guys. And thank you guys so much for listening. We'll see you next time, and looking forward to it. See you guys. Bye.
Bye.